Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Welcome back to the Deep Dive. Today, we're tackling something fascinating, almost like a superpower in cancer cells, specifically melanoma, and its ability to, well, completely change its identity like a chameleon.
Right, becoming essentially invisible or impervious to standard drug treatments. It's a huge problem.
But here's the twist we're exploring. What if that very identity change, that superpower, actually creates a fatal flaw, maybe something linked to iron metabolism?
That's exactly what this new research suggests. For years, the focus was nearly all on gene mutations, But this study from VIB researchers, published in Nature Metabolism, it really shifts the spotlight. It points to a deeply conserved metabolic process driving this chameleon act. And yeah, iron handling seems to be right at the center of it.
So the key to beating these incredibly resistant cells might not be in the DNA code, but maybe in controlling how they manage iron. Our mission today is to unpack that.
Yeah.
We need to understand how melanoma cells pull off this identity switch and look at the specific molecular machinery, the sort of traffic control system for iron.
And critically, how knowing this might open up totally new ways to target these resistant cells.
Right, almost like turning their own defense mechanism back on them.
Precisely. It's a story about how cells adapt, but also about the potential weaknesses these adaptations create.
Okay, let's start with the switch itself, this phenotypic plasticity. The sources talk about two main states these melanoma cells flip between.
Yeah, they're generally referred to as the MEL state and the MES state.
MEL.
Melanocytic. Think of this as the more standard cancer cell state, if you will. They're highly proliferative, busy, dividing, and growing. And importantly, these MEL cells are the ones susceptible to our current therapies. That's where treatment often works initially.
That's the state we can hit effectively. But then the drugs apply pressure.
Exactly. And some cells manage to switch. They transition into the mesenchymal-like, or MES state. This is a whole different beast. These cells are invasive, they move around more. Crucially, they are highly drug-tolerant. They're the reason treatments often fail long-term, leading to recurrence and metastasis.
They basically put on armor against the chemo.
You could think of it that way, yeah, a protective state.
And the big revelation here isn't just that they switch, but how. It's not just genetic reprogramming anymore.
Well, the genetics are involved, but this study highlights that metabolism, specifically iron metabolism, might be a key driver enabling the switch. It's upstream.
Okay, so it's about the cell's internal logistics, how it manages resources like iron.
Exactly. And it involves communication or crosstalk between different parts of the cell machinery, organelles talking to each other.
So if iron is the cargo, what are the key cellular structures involved? The warehouse and the power plant, perhaps.
That's a great analogy. The two main players here are the lysosomes and the mitochondria. Lysosomes act like the cell's recycling and storage center. Iron comes in, gets processed, stored there.
The warehouse.
The warehouse. And mitochondria are, of course, the power plants. They absolutely need iron to generate energy for the cell to survive and function.
Makes sense.
Yeah.
So how does the iron get from the warehouse, the lysosome, to the power plant, the mitochondria? Is there a direct route?
There needs to be. They found these physical connection points, literally like bridges or tunnels between them. They call them mitochondrial lysosome contacts, or MLCs.
MLCs, okay.
These contacts are essential for moving cargo, like iron, efficiently over that short distance. Think of it like a dedicated loading dock connecting the two.
Got it. So in the resistant MES cells, something goes wrong with this delivery system. The loading dock breaks down.
That's what the researchers found, a clear defect in iron transport in these MES cells. The MLCs seem disrupted, or the transfer mechanism isn't working right.
And the result?
Iron piles up inside the lysosomes. It gets trapped, sequestered in the warehouse.
Wow, the power plant.
The mitochondria. They end up starved of iron. Their iron levels drop significantly, and this directly impacts their function. Mitochondrial activity goes down, which fits perfectly with what we know about MES cells. They have a lower capacity for energy generation through respiration. They're running on low power.
Okay, so the traffic gets jammed. Why? What specific molecule or machine is supposed to be running the show at that loading dock? And what happens to it when the cell shifts identity?
This is where it gets really specific. The key player they identified is an enzyme called BDH2.
BDH2.
In the drug-sensitive MEL cells, BDH2 is abundant, and it's located right there at those mitochondrial lysosome contact points.
Its job?
Its job is to produce a small molecule called 2-sulfide-dihydroxybenzoic acid. Let's call it the iron ferry molecule, 2-5-DHBA.
The iron ferry, I like that. So BDH2 makes the ferry.
Exactly. And this iron ferry, 2-5-DHBA, acts as what's known technically as a cytophore.
Cytophore. That sounds familiar. Isn't that something bacteria use?
Precisely. That's the fascinating link. It's an evolutionarily ancient mechanism. Bacteria use cytophores to scavenge iron from their surroundings, basically stealing it to survive. These melanoma cells are using a similar, likely related internal mechanism. The iron ferry grabs iron inside the lysosome and transports it across to the mitochondria.
Fueling the power plant?
Supporting the cell's energy production, yes.
Okay, so the sensitive cells use this ancient bacterial trick with the BDH2 enzyme making the iron ferry. It sounds incredibly important for the cell. So why on earth would the melanoma cell shut it down when it switches to the resistant MES state? It seems counterintuitive.
It does, but it seems to be a trade-off, a calculated risk, perhaps. When the cell adopts that invasive, drug-dodging MES phenotype, it significantly reduces or completely loses the BDH2 enzyme.
Which means?
No more iron ferry production. The ferry service stops. And without the ferry, iron gets stuck in the lysosome. The mitochondria stay starved.
But this somehow helps the cell become drug-resistant.
It seems linked to that lower metabolic state, that laying low strategy of the MES cells. They sacrifice peak mitochondrial efficiency maybe for stealth and survival against drugs. It's a compromise.
But like any compromise, there's a downside. This intentional iron mismanagement trapping iron in the lysosome, it creates a new problem, doesn't it? An Achilles heel.
A major one. This is where the concept of ferroptosis comes in. That buildup of iron in the lysosomes isn't just sitting there passively.
It's reactive.
Highly reactive. It's redox active iron. It essentially primes the MES cells for a specific type of cell death, like leaving explosives lying around. That specific death pathway is called ferroptosis.
Right, ferroptosis. We hear that term more often now. Can you quickly define it for us? It's not the usual programmed cell death, is it?
No, it's distinct. Ferroptosis literally means iron-dependent cell death. It's triggered by the catastrophic oxidation of lipids fats in the cell membranes. Think of it as overwhelming cellular rust, driven by that excess reactive iron, causing membranes to just fall apart. It's messy and explosive.
And because the MES cells have deliberately stockpiled this reactive iron in their lysosomes,
they become uniquely, exquisitely sensitive to being pushed into ferroptosis. They're sitting right on the edge.
The researchers actually showed something quite clever about this related to BDH2, didn't they? A kind of protection paradox.
They did. They showed that if you take those resistant MES cells, the ones lacking BDH2 and vulnerable to ferroptosis, and you either restore the BDH2 enzyme or just give them the iron ferry molecule, the 205-DHBA.
What happens?
They become protected from ferroptosis.
Protected? Why?
Because the iron ferry starts running again. It clears that dangerous buildup of iron out of the lysosomes, moves it to the mitochondria, and basically disarms the ferroptosis bomb, restores the balance.
Wow. So the very thing that helps them resist drugs losing BDH2 and trapping iron makes them vulnerable, but restoring it protects them again.
It's a fascinating dynamic.
And this protection mechanism, this BDH2 iron ferry axis, it has huge implications for how cancer spreads, right? For metastasis.
Absolutely critical implications. Their experiments using mouse models showed this system plays a key role during metastasis. Specifically, it seems to shield these MES cells from ferroptosis while they're traveling in the bloodstream.
Which is a really hostile place for a cancer cell, I imagine.
Extremely hostile. High stress, lots of oxidative damage. Most circulating tumor cells actually die, many likely via ferroptosis. But by managing their iron carefully using this BDH2 pathway, the MES cells that retain it, or perhaps reactivate it, can survive that dangerous journey through the circulation.
Giving them a much better chance of reaching a new organ, like the lungs in their models, and setting up a secondary tumor.
Exactly. It enhances their metastatic potential by protecting them during transit.
So this isn't just about iron moving from A to B. It's part of a larger interconnected system. You mentioned a feedback loop earlier.
Yes, it all ties together quite neatly. The BDH2 iron ferry system feeds iron to the mitochondria. Healthy mitochondria produce energy efficiently through oxidative phosphorylation.
Okay.
That energy, in turn, is needed to power other cellular machinery, including a crucial pump in the lysosome membrane, the VAT-PACE proton pump.
And what does that pump do?
It maintains the highly acidic environment inside the lysosome. That low pH is essential for the lysosome's function. And here's the loop. that acidic pH is also required for those mitochondrial lysosome contacts, the MLCs, to form and remain stable.
Ah, I see. So let me try to trace that. The iron ferry, BDH22-5-DHBA, gets iron to the power plant mitochondria.
Right.
The power plant makes energy, which runs the security system, the acid pump. And the security system, acidic pH, makes sure the loading docks, MLCs, are stable so the ferry can keep running.
You've got it. It's a self-sustaining metabolic circuit in the healthy or MEL state.
And that whole circuit collapses when the MES cells lose BDH2. The ferry stops, the power plant sputters, the acid pump might weaken, destabilizing the contacts.
That seems to be the core logic emerging from the study. It's a cascade effect triggered by shutting down that initial BDH2 step.
Now, the researchers were careful to point out a limitation, weren't they? Even when they put BDH2 or the iron ferry back into MES cells, something wasn't fully restored.
That's right. While restoring the BDH22-5-DHBA axis largely corrected the iron flow defect and protected from ferroptosis, it only partially rescued mitochondrial respiration.
What does that imply?
It suggests that while this BDH2 iron pathway is clearly a major player in the metabolic state and the ferroptosis sensitivity, there are likely other factors involved in the full metabolic profile of MEMEL versus MES cells. Other metabolic adaptations or maybe signals from the tumor microenvironment also contribute.
It's rarely just one switch in cancer, is it?
Almost never. But identifying such a critical node like BDH2 is still a huge step.
Absolutely. And the potential here is significant. Professor Patrizia Agustinus, one of the senior researchers, was quoted saying this work opens up innovative strategies targeting iron balance and this organelle communication to fight resistance.
Yes, and the implications might even go beyond cancer. This kind of iron mismanagement, problems with lysosomes and mitochondria talking to each other, these are also features seen in aging and several neurodegenerative diseases. Understanding this mechanism could potentially offer insights there, too.
That's really broad potential.
Okay.
Let's try to synthesize this. We've journeyed through how melanoma's drug resistance involves a dramatic metabolic shift.
Specifically, shutting down the BDH2 enzyme and its iron ferry molecule, 2,5-DHBA.
Which jams up iron traffic, trapping iron in the lysosomes and starving the mitochondria.
But that very act, that defensive maneuver,
Creates this profound vulnerability, a sensitivity to iron-dependent cell death, ferroptosis.
exactly. The defense creates the weak spot.
So this prompts a really provocative thought, doesn't it? Maybe the standard approach, trying desperately to stop cancer cells from ever switching to that resistant MES state, isn't the only way.
It certainly makes you reconsider. What if instead we let them switch, or even encourage it, and then once they're in that iron-loaded, vulnerable MES state, we hit them hard with drugs designed specifically to trigger ferroptosis.
Use their own internal iron bomb against them, target the resistance mechanism itself.
It's like metabolic warfare, weaponizing the cell's adaptation. It's a completely different angle on therapy.
A fascinating prospect, turning the cancer shield into the weapon that destroys it. Definitely something to think about regarding the future of cancer treatment.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.